discussion of the metabolites related research which are just can be used for the different purposes and can be used for the different medicines also. Tea is itself is a medicine. Their own sites, education portals as well as the SWAYAM portals, and it will be just like for everyone. So that's the reason that we would like to give you the course material after splitting of the different courses. So that was a time consuming process, and whatever the slides we are giving, it will be uploaded on the SWAYAM courses also. So Ministry of Education program is really very ambitious. They want to benefit each and everyone whoever is sitting anywhere here. They can also take the benefit of this thing. So yes, I am starting that is the tutorial in which we have to explain. I will go a little bit slow right now because in the morning there was an issue of the speedy presentation. So I thought that I have to cover each and every point. So I was a little bit speedy in the morning. So first point here come up that what is the meaning of the metabolic control analysis and where we need this metabolic control analysis. So try to understand that this is just like a framework, structure which help to understand the control of the fluxes in the metabolic pathway at the molecular level. So now I would like to elaborate this thing in this way that a biosynthetic pathway has many reactions. Okay, every reaction has its own speed. So this metabolic control analysis help to understand that which flux, which reaction is the key determinant for that particular biosynthetic pathway. So basically, we have to use whatever the existing knowledge available online about the biochemical pathways and the network, which can be used to understand what is the speed of the different biosynthetic reactions in the whole pathway. So there are the 2 points, that is the steady state fluxes and metabolic concentration. These are the 2 points parameters. We will discuss these 2 parameters in the detail. These 2 parameters influence the enzyme property and other parameters of the system. So there is a direct correlation between the steady state flux, metabolite concentration and the enzyme activity. So we try to understand how these metabolite concentration, steady state fluxes influence the enzyme property also. And this MCA helps us to know that how this metabolic effect, when you manipulate the different biosynthetic pathway, so a upregulation of a particular metabolite or downregulation of a particular metabolites, how they influence the overall metabolites which are present in the cell. So, and our focus is about that we try to, how we control this pathway, and that is the most important part, because we have to identify the key limiting steps of those biosynthetic pathways. If you know which pathway, which reaction, which enzyme of a particular steps of a biosynthetic pathway is the key determinant. So you can use that one for your different experiments also. Now, the questions come up that why you need the MCA actually, metabolic control analysis, and what is the importance of this one. So MCA is basically, there are a few gaps in the MCA. The major gaps in the MCA is that we know only limited number of the metabolites, and we have to determine each and every metabolites with highly sufficient accuracy and the precision, because it's a time consuming process and it requires this is the major points we have to elucidate any biosynthetic speed of any biosynthetic pathway. And the complete description of the entire system at the atomic level is really undermined. We have to generate the data, we have to compile whatever is already exist to understand this thing in a comprehensive way. And yes, when we are trying to understand the MCA, we must know what are the different enzymes which are participating in this biosynthetic pathway and what is the importance of those enzymes in this biosynthetic pathway. Basically, there are the two different frameworks which are used for this particular understanding the behavior of the different steps of any pathway. The first one is the biochemical system theory that that is not a point of attention today because we would like to go ahead with the MCA. MCA is really starting with this thermodynamics and the kinetics actually. So let me take this example and you can understand how these flux are regulated actually and how a enzyme decide that now this reaction is going moving direction, how this reaction is going in the backward direction. So what is the K factor which determines the direction of any steps in a biosynthetic pathway? So let me explain with a small example. Here you know every cell has the different biochemical reaction and thermodynamically they are explained here. So thermodynamically we are going with the delta G actually. Delta G is something that is giving us gives free energy thermodynamically. So if you understand the delta G dot, so it is the minus RT in K, where K is an important parameter that we have to discuss right now. But before that, that is equal to the delta H minus T delta S. So you know this is thermodynamics reaction and all these parameters directly or indirectly influence gives the energy that is required to start the reaction or they release the energy when reaction is automatically come up in the same. So when delta H is the 0, enthalpy is the 0, then K is the dependent upon the temperature actually. So yes, K also depends on many other parameters just like the ionic strength, water activity, pressure and the wide variety of the variable also influence the value of the K. Now, when there is an equilibrium between the substrate and the product, so net flux is also the 0 actually. So nothing is going from the substrate. When a substrate is going to convert into the product, after formation of the product and equilibrium exists, now there is no more substrate molecule are going and convert into the product. And no product molecule is going back and reverse in the reverse mode and produce the substrate. So that means that flux is the 0. So flux is when for the free energy that we define departure from the equilibrium, when there is equilibrium that can be deviate due to the some reason. So free energy is a parameter to calculate how much it is deviating from the equilibrium. So this equilibrium is just like the ratio. Chemical equilibrium is, if we define theoretically, so it is just like a ratio of the chemical activity of the product as well as the substrates, reactants. So you can say here K is the chemical activity, so it is just the activity of the product divided by the activity of the reactants. So this is just small, but you will understand very well. So now, these activities are basically cast in the form of an equation. What is the chemical activity which is happening in the reaction? So this is equal to the epsilon C. C is the concentration, epsilon is specific coefficient with respect to the concentration time. So in the ideal condition at the standard level, this value is equal to the concentration. That is the epsilon C is equal to the total concentration that is present at a particular site. So this equilibrium is basically also expressed in the form of an activity ratio. So this activity ratio is expressed in the form of P is equal to AP divided by the AS. A is the activity of the product, AS is the activity of the substrate. So the equilibrium is depends on the ratio of the activity of the product as well as the activity of the reactant. And finally, if there is any deviation from the equilibrium, so you can calculate here if this K, this T divided by the K. K I told you that is associated with the equilibrium. T is the activity ratio. So T divided by the K is less than 1, then reaction is proceed in the forward direction. If this equation is opposite, means T divided by the K is greater than 1, then it goes from product to the substrate in the reverse mode basically. So basically this free energy of this reaction is given here. So here there is an example, G1 is equal to this one. So G2 is equal to this one. So finally you can understand that there is minus 7 kilocalories per mole is the energy that is released here. So it means now we have to decide this is the delta free energy. So this free energy is drives some reactions. So net flux of the any reaction, flux means the speed, pace by which the reactant is going to convert into the product. So net flux of the reaction thus depends on the degree of the departure from the equilibrium. So when there is no equilibrium and some equilibrium is disturbed, then this flux come up in the picture. So it represents, yes, this reaction is going in the forward or this reaction is going in the downward direction. So let me explain this whole concept with a small illustration here. So these are the substrate means substrates, P means product. This is a multi-step process and finally this substrate is converted into the product by many steps. From the B, then followed by the C, followed by the D, subsequently it produces the product that is the P. And remember, these 4 different reactions are catalyzed by the 4 different enzymes here. E1, E2, E3 and E4, they respectively catalyze the different reactions from the substrate to the product actually. So this flux is basically depends on the only the concentration of the metabolite, that is the P and S only. So overall concentration of the enzyme, their intrinsic catalytic powers and their overall equilibrium constants, these parameters we have to calculate. And at a steady state when there is nothing is going on, you have to freeze the reaction, when you have to try to capture the glimpse of the reactions. So at each rate at each step is the same actually. So in the steady states, all the rates are almost the same, starting from the S to B, B to C, C to D and D to P. Conversion is the same and you can go back in the P to D, D to C, C to B and B to S actually. So this equilibrium ratio is symbolized here by the T and ratio is defined here. It is a unity at the equilibrium. So net flux is basically maintained by the inputs of the substrates. If substrates is not available, so there is no flux basically. So and where from these substrates come up basically? These substrates come up from the outer part, exports. They are coming from the cell membrane, they go inside and they are available for the reactions. So transports or transporters gene are also very important. If you are not considering the transporter genes or enzymes and you are just doing the biosynthetic pathway, so might be you will get something in a misinterpretation of the whole metabolic pathway. So you have to consider how much metabolites are imported inside the cell from the outside, from one organelle to the cytosol, from the cytosol to the different organelles also. So for the simple scheme, we can define this particular concept that is representing here as a J is equal to FKS and 1 minus T. Just this is one more equation, otherwise it is complete. So in the linear scheme, you can define J. J is the flux basically. J is S minus B because and J is minus B minus C. So I am going back B and C and D. So J, S, B, S and the B is the first steps and then J is equal to BC. So BC. So both are the equal basically, but in the linear fashion. But these flux are basically when you are considering all the different steps in a particular biosynthetic pathway, so you have considered J bracket S and the P. Now you have to consider S to A to B, B to C, C to D, this way also. So this is the elaborated statements which reaction which including all the different steps within this biosynthetic pathway. Now same enzyme basically is going forward and going reversely. So yes, that enzyme activity of that enzyme can be increased very low value to a very high value also. So flux J increasing hyperbolically when you increase the activity of the enzyme. And because active is the enzyme is something which is forwarding the metabolites from the one step to the another next steps. So if you increase the concentration of the enzyme, so definitely the flux is also increased. Now, if you are down regulate those biosynthetic pathway and you just put one enzyme at a very low concentration, so yes, then less amount of the metabolites is going to convert into the product. The reactant is going to product. So flux is also down in that case. So enzyme concentration increase and the decrease directly influence the flux of any biosynthetic pathway of any steps. So it can become very capacity of the other enzyme also matters. A enzyme, how much the different reactant can accommodate, and there is a few another parameters we have to understand. So this is a normal illustration in which flux is present with respect to the enzyme concentration. In the first table you can see here enzyme activity or balanced concentration of the enzyme with respect to the flux. It is highly balanced. You are increasing the enzyme concentration and flux is also going to increase. This is a hyperbolic. After some time there is no change in the enzyme activity or there is no change in the flux also. If you increase the enzyme activity, even then your flux is remain constant. But if you add some inhibitors, so what will happening here? So this whole concept is going down. You increase the concentration of the enzyme, but if you add the inhibitor, so after some time your whole flux come at the zero. So it means you seize the whole biosynthetic pathway by using some inhibitors. So now there is no reactant is available which can proceed and convert into the product. So this thing help us to know that how these inhibitors can manipulate or can fix the stop the biosynthetic pathway and kindly at the zero values. So let me take an example for all of you. If you are working on the biosynthetic pathway, so you have to understand many concepts here. So I am here taking an example of the glutathione plant. Glutathione is just like a small molecules, but it is very useful. It is useful during the abiotic stresses, biotic stresses. It is also used for the xenobiotic detoxification concept. Then it is also used for to provide the tolerance during the heavy stress conditions. So yes, this metabolites is routinely used. You know about the disulfide homeostasis, sulfur homeostasis and disulfide reduce and the oxide metabolites. So this glutathione is also providing the redox homeostasis to the cell actually during the stress condition and during the normal condition. And this is the synthetic pathway of this glutathione. It is starting with the sulfur, and yes, this synthetic pathway has the gamma glutamine cysteine synthetic enzyme. Another enzyme that is the most important is the glutathione synthetase. So these are the GS is present here down. GS is present here, and you can find out this delta ECS. This is just the upper reaction of the whole biosynthetic pathway. So these two pathways are more important for this manipulation if you want to increase or decrease the glutathione concentration inside the cell. And remember, these two, they are the few more also require the ATP. So basically, broadly, it is classified that whenever any step is using the NADPH, ATP, so they are the rate limiting steps. But every time it is not accurate. So here, the two enzymes are the K regulatory enzyme, which is manipulated by in many pathways, and they try to accumulate the glutathione, and they also try to accumulate the glutathione in the vegetable as well as the fruits also. So I am coming here now. What is the regulation? Because we are considering our whole investigation based upon the two different enzymes. That is the GS is one of them, glutathione synthetase. So here you can find out these are the regulations, enzyme catalysis. So they catalyze the reaction. Both these two enzymes catalyze these two reactions with a very high equilibrium constants that I told you. So K equilibrium, that K in this we discuss here K. This K. So this K has very high value here. it is more than 1000 times. So now second is that what is the regulation under the low glutathione demands. If there is a low demand of the glutathione, so how it regulate? So we try to inhibit the glutathiones, this delta ECS, to prevent the excessive this cysteine accumulations, and now no reactant is available. So finally they maintain the steady states in the normal condition also. So we can regulate the low glutathione concentration also here, and this glutathione and the cysteine regulation we can manipulate by inhibition of the glutathione and the cysteine. These are the reactant basically. Glutathione and the cysteine. Glutathione is the product, cysteine is the reactant. So if you inhibit of the whole biosynthetic pathway by these two metabolites, so this ATP sulfur hydrate, ATPase and the sulfate transporters are regulating by the glutathione and the cysteine. And the regulatory feedback, both the metabolites, glutathione and the cysteine, also provide the regulatory feedback here. So they inhibit when their concentration is over-accumulated, so they inhibit the whole concept. So finally it was documented that this is a K enzyme which is a rate limited steps in the glutathione synthesis under the normal condition, not under the altered condition. So we can extend it with the high demand of the glutathione. So we assumed that we have to go ahead to manipulate this biosynthetic pathway, these particular steps, if we really want to improve the glutathione in a particular condition, that is the heavy metal exposure, biotic stresses, other abiotic stresses also. So that's why now on the basis of all this biochemical analysis, you can understand that which enzyme is giving the better information with respect to the flux analysis. So here we finally identify that this enzyme is the K regulatory enzyme, and we have to proceed with respect to this particular enzyme also. So now how you identify the different steps basically? These are the biotechnological application of this altered glutathione, our accumulation of the glutathione. So basically this enhanced amount of the glutathione is also the source of the formation of the phytochelating components. So this phytochelating is basically used for the heavy stress conditions. So that's why it is something we can go ahead. And goal of this study you can write down in your project or the small report if you want to improve the bioremedial strategies, and we can find out how this accumulate toxic metal ions influence the biochemical redox homeostasis of that particular plant under the stress condition. So that's why we come up with such type of the framework for your better understanding. You know how to draft the objective, how you understand the thing. So for that we have to go ahead with our expression of the glutathione, and that is really a challenge. And results, our expression increase the glutathione level with no clear correlation to at the enzyme level. Yes, they increase when you manipulate this particular enzyme, our express, and then you identify that glutathione concentration is going to increase, but there is no influence on the activity of the enzyme. So there was a doubtful, if activity is not increased, then how this flux is going to increase in this particular situation. The second point is that it means maybe there is activity is not increased, so maybe this pathway is not complete yet not, and we have to find out. So limitation in the glutathione pathway is that we focused only this synthetic reaction, and we are ignoring the consuming reaction where this glutathione is going to downgrade or use as a substrate. So maybe there is chances that when you express an enzyme, so expression is increased, but enzyme reaction are the same, and you are getting the same metabolites, but the degradation of the utility of the glutathione is going to reduce. So that's why the glutathione concentration is upregulated during stress condition under our expressed situations. So now that's a reason that this way you can understand that we have to consider some other parameter, hidden parameters, which is essential for all of us. So such type of the information you can only extract if you know about information about the MCA. MCA is basically, it can be used to identify the control steps basically. So qualitatively describe the substrate flux is response to change in the system parameters. So you have to, substrate flux is something which can be changed with respect to the system parameter. So if you manipulate a few parameter, so you can find out that substrate flux is also going to increase and decrease. This thing also help to identify the enzymatic targets for the different disease, including the cancer, drugs discovery, as well as the plant-related disease also. So you can identify how these enzyme targets happen in that particular case. Sometimes if they, in an infection, biotic stress, some enzyme are targeted, and due to those targets, they identified altered level of those biosynthetic pathway. They basically seize the whole biosynthetic pathway. So try to understand, the same thing happened in many cases. A single enzyme can influence the whole disease in the animals also. The same situation is repeated in the plants also. The another important point of this MCA is that it links the biochemistry with the genomics. So genomics is giving the detailed information about the genome structure of any organism. So, but this biochemistry is giving why there are so many changes happening at the biochemical level. So genotype and the phenotypic correlation is possible when you are working with the MCA here. The another point is that, if you are looking only for the one enzyme for the flux analysis, so might be you are a little bit wrong in this interpretation. Sometimes the whole flux is not controlled by the one enzyme, it is controlled by many other enzymes also. So many times you saw the papers, they are coming, our expression, down regulation, silencing of the particular enzyme, and they are altered the whole biosynthetic pathway, and they show that someone is increased and someone is decreased. So that only due to the one enzyme is it not possible, but maybe our accumulation of that product catalyzed by that particular targeted enzyme may be used as an activator for the successive steps, and that's why they are getting better accumulation of those metabolites. So yes, we have to also identify, this MCA is also very important if you want to identify some specific targets for genetic, chemical and the dietary intervention. You know, some people have some specific dietary issues, allergy. So when you go and find out there is some enzymes which are not capable to digest those foods, specific foods actually. So if we are working on the MCA, we can identify which enzyme is responsible for such type of the metabolic intervention, so that you can improve the digestion system of that patient. So I am just explaining a small story. You know about the insulin. So when insulin was not discovered, so it was a mysterious disease actually. So they identify so many small kids. They stayed they are just like the coma. They are going to enter into the coma states. Very less weight, underweight, malnourished person, because whatever they are taking the foods that was not digested in their body properly, and high level of the sugar is something that. So when this insulin, when the scientists cite insulin, so they first of all feed the insulin injection to those girls actually, and those girls within the 40 days come back from all the diseases symptoms, and they become the normal behavior. So this insulin was sold to the market at 1 rupee patent only, and that's why understands they chase the whole concept of the diabetes. So that's why I would like to say that this MCA is also a component which can help to identify if an enzyme is not working very well, and that is responsible for a particular metabolic disorders. So if manipulation of that enzyme could helpful to provide the positive influence after manipulation of those biosynthetic pathways. So this is another case where we have to go ahead with the, it was a traditional view on the rate limiting steps of the glycolysis. So glycolysis is, you know very well, and here you will identify that everyone is identified what are the genes which are involved in the glycolysis. And you know very well rate limiting steps by using the qualitative criteria, so that according to that one enzyme governs the whole metabolic flux. So let me discuss, is it okay or not? So this concept is oversimplified here to understand the complexity of the metabolic pathway. So here case study is the glycolysis. So traditional identification of the K enzyme are those one: hexokinases, phosphofructokinases, pyruvate kinases. These are basically tagged as a rate limiting steps based upon what type of the approach is. These are the old approaches we adopted actually. So theological approaches, then thermodynamics approaches, and the kinetic approaches. Finally, we come up that these are the few enzymes which are essential for all of us. There is another approach that was used as an enzyme cooperatively approach. So enzyme cooperative approach go ahead. A few enzymes are altered allosterically and they influence those biosynthetic pathways. That is the PFK1, PYK, and this HK, hexokinase, is a regulator which product inhibitor of the glucose 6-phosphate and ADP reaction enzyme also. So now according to these different approaches, we have enough information about this biosynthetic pathway. But there is another point. There is a little bit variation in the fructose 6-phosphate and the fructose 1,6-phosphate concentration. Suggest us that they also control the PFK1. So now, what is the limitation of the traditional adaptation basically? Yes, we ignore the glucose transport gene actually. So now those things also come up present. So if you are not getting the glucose from that organelles, from the outside or from some transporter, so how this glycoreaction happened actually? So then there is a doubtful key, it was the linear and the previously it was discussed that this pathway is the linear and branchless, despite the presence of multiple branches, hypothetically. So we assigned full control of the keys which regarding contributes from the other enzyme and the transporter. So try to understand, when you are discussing about any biosynthetic pathway, so you have to cover from their synthesis, their transports, where they are going, how much is breakdown, what is the final product. So you have to take care of all those components. So this is the illustration. Here it is the glucose, then glucose 6-phosphate, fructose 1,6-bisphosphate, glyceraldehyde 3-phosphate, then 3-phosphoglycerate, phosphoenol pyruvates, and the Krebs cycle. So, but whenever you are finding the ATP, so we are randomly assumed that these are the major pathways, major enzyme catalyzed reaction which we need to consider for our. But this investigation excluded the transporter gene also. Transporter genes means they are something which are providing which transport the glucose to the cell, and alternative branches also. If you consider, a new additional control point emerges. Metabolic control analysis means you are controlling the different biosynthetic pathway. How you control a biosynthetic pathway? What are the different stages where you can control the different biosynthetic pathway? If you have the overview, if you are considering all the factors, so you can control. Otherwise, it is really very difficult to say that this our accumulated product is due to only a single enzyme. So now, glycolysis control in the practice. So experimental refinement was carried out, and with the help of a few inhibitor titration turnout approach for the lack of specific inhibitors. So that's why it was carried, titration inhibition was carried out. Second, let me take an example of this one. Iodoacetate identified GAPDH as a rate limiting steps enzyme in the Streptococcus lactis and the Cerimoris. So it was by titration inhibitor titration method, it was identified that a few other compounds are also working as an inhibitor. Now, conserved nature of the glycolysis across the species. So what happening actually, this glycolytic pathway is highly conserved evolutionary with minimum variation in the enzyme. Differences in the enzyme property. Substrate property is also differences in the glycolysis. In certain biosynthetic pathway which makes the glycolysis a therapeutic target for the disease like cancer and the parasitic infections. So you know, glycolysis is highly active. Cancer cell needs so much energy for the rapid cell divisions. So sometimes the high substrate specificity in certain pathogens make glycolysis just like a therapeutic targets. Now the application of the glycolysis control. So clinical relevance is also mentioned here. Inhibiting the glycolysis can be combat with the cancer and the human parasitic disease. So whatever some parasites are depends upon us for the sugar actually, glucose. And cancer cell, we can manipulate the cancer cell if we inhibit the targeted glycolytic pathway in only the cancer cell. Then there is no need for the medicine. We have to identify the inhibitors which can do this thing only in the cancer specific cell. So biotechnological approach that we need to go ahead. Glycolysis produce many side products like the ethanol, CO2, lactic acid, which can be used for the different purposes. Beverages, bread, and the cheese. So increasing glycolytic flux can optimize the production time and enhance the metabolite concentration also. Physiological insight, if you think so, this muscle glycolysis can increase the 100-fold during the activity with minimum change in the intermediate metabolic concentration. So yesterday I read an article. If you are doing the 15 minutes exercise, so your brain map is changed. And if you are relaxed, so your brain map is different, and someone doing the exercise, so their brain map is totally different. So try to understand a 15 minutes exercise is enough to change the whole profile of your brain. But during exercise, your body requires so much glucose, and every cell has the glycolytic pathway. So during the exercise, glycolytic is taking care of everything, while all other metabolites are present as a same situation. So try to understand if you go ahead with this pathway, so you can do the miracle. Now what are the advanced strategy which you can use for the in the glycolysis? You have to design. So this is the target. If you are working on the similar things, so you can go design metabolic intervention to manipulate the flux and intermediate metabolic levels of a particular pathway in a particular plant. So now this way you can make the whole title of your work. Approach you can use just like a quantitative framework for distribute the control across the multiple pathways below our simplified rate limiting steps. So basically we are saying that this pathway has 2 steps, this pathway has the 3 steps. If you are working with this MCA, so you can find out the whole quantitative framework which explains all the different control points in that particular pathways. So now I am coming with another part, that is the flux and concentration. So how this flux and the concentration are associated with each other. So FCC is just like a flux, and the concentration control coefficient is quantify the contribution of each enzyme into the pathway. So previously we are saying that only one enzyme is the rate limiting enzyme which catalyze a particular step, so we can manipulate them. But here we are mentioned that yes, we have another approach which can quantify the contribution of each and every enzyme which are present in that particular pathway and which are involved in that flux. So this is the formula of this is the concentration control coefficient flux, and this is the flux along with this enzyme concentration. So you have to use the 2 parameters. First is the overall flux of that biosynthetic pathway, and second is the different enzyme and their concentration. So yes, flux control is distributed across the multiple pathways, so that's why it is something that can give us better information about the contribution of the different enzyme with respect to that particular control pathways. This is something, the maximum value of this thing is always equal, and if it is never 1 actually, it is always less than the 1. So this is the summation theorem which is used for the flux and the control concentration control coefficient, that is the FCC, and it is the sum of all the enzymes. You have to calculate individual and then you sum up this value, and it never crosses the 1. It is always less than the 1 because sometimes some factors are not possible. So this is the flux control coefficient, and this is the flux along with the enzyme concentration. So here you can find out flux control coefficient. So it is a linear metabolic chain where all the enzyme flowing the Michaelis Menten kinetics, and finally the flux control coefficient is changed for a high value at a low enzyme concentration and approaches zero when the concentration of the enzyme exceeds the capacity of the remaining enzyme of the pathway. So it also becomes the zero along with this one. If you increase the concentration, in the 10th standard or before 10th standard, you read about many equations. If one person is doing this work in the 10 days, so how many 5 persons do this work in how many days? So if you add 100% do, so it will be again the 5 days, because many of them will be shifted into another assignment. But here what happening actually, if you are increasing the number of the enzyme, so the flux is going to down, and when more enzyme is present more as compared to the reactant, so flux is going to become the down and it become the zero. So this is the experimental strategies for the MCA that you can go ahead, methods to determine the this MCA, that these are the 5 different methods which we are generally mentioned to determine age. The first one is the heterokaryon and the heterozygote formation. Second is the titration of the flux with specific inhibitors. We discussed it a little bit in the previous slides also. Elasticity analysis, then mathematical modeling, in silico biology you can say, in vitro reconstitution of the metabolic pathway, genetic engineering to manipulate to in vivo protein level also. So we are working on the genetic engineering to manipulate in vivo protein level. One of my students is working on this concept. So now the classical Mendelian genetics, this was the first metabolic pathway where flux control coefficient was experimentally determined, and the organism was the Neurospora crespa. Approaches, they use the mycelia formation formed by the mixing spores with different genetic composition to study the enzyme activity and their influence on the flux. So here they manipulate the different enzyme in the different mycelia, and they mix together and they try to understand what is the influence of this thing on the whole metabolic pathway. So experimental was set up by this way. So mixed spore with the gene of the wild type and the mutants of the arginine biosynthetic pathway. Basically arginine biosynthetic pathway was first of all MCA was reported with this particular pathway. So these are the 4 different pathways. That is the acetylide, aminotransferase, ornithine trans, carboxymylase, arginine succinate synthase, and arginine succinate ligase. So finding of this work was that enzyme activity, so flux control coefficient between 0.02 to 0.2, indicating that the total flux is in between the 2 to 20% only. It was not more than that. And one of the K enzyme was identified as a K control over the arginine biosynthetic pathway. 70% of the total flux was the undermined, because we go only 2 to 20 maximum. And potentially due to the a few synthetase and the enzyme also, transporter inhibitors and the phosphatase synthetase, because we are not covering those one here. We make the mutants with respect to those particular assignments. So flux control in the genetics can be highlight the significant flux control is not always exerted by the individual enzyme. Even their activity level is also very low. Implication of the MCA is that it is below the rate limiting steps. You not only have to identify the rate limiting steps, but you also have to identify some factors which are below the rate limiting factors, and they are controlling the whole metabolic pathway, that specific pathway. So this is the second thing that we discussed a few slides before also, that is the titration of the flux with inhibitors. So control of the oxidative phosphorylation. Let me take this example for this better understanding of this methods. So it is a method to evaluate the flux control coefficient by using some specific inhibitor to reduce the enzyme activity. So application of this one is that this is an enzyme, that is the OX, oxidative phosphorylation, due to the availability of the potent inhibitor for many of the enzyme and the transporters. Primarily, because you know about the cyanides and many other poison. So these poison are basically inhibitors of some enzyme. So oxidative phosphorylation is a K reaction in the biological system. If you inhibit the oxidative phosphorylation reaction, so you are seizing the whole system basically. And this is not only a inhibitor of a single enzyme in the transporter. Oxidative phosphorylation is just like influencing many potential inhibitors which influence the enzyme and the transporters. So now we have to divide this oxidative phosphorylation in the two parts. First is the oxidative system and second is the phosphorylating system. Oxidative phosphorylation system may we have to find out the substrate transporters which are used. Second is the Krebs cycle enzyme, respiratory chain complex. So now here you have to, oxidative phosphorylation is, if you consider it is a small step, so might be you are the wrong here. We have classified many components. We have to check each and every factor to find out that how this inhibitor influence the different component of the oxidative phosphorylation. So phosphorylation system here, ATP and the ADP translocases, phosphate transporters and the ATP synthase. So these systems are interconnected also by using the proton electrochemical gradients. So if you manipulate one, so sometime you will find out they also influence the other factors. And finally, they are sometimes manifold down regulates the thing, and you think that only one enzyme. Multiple enzymes are involved for the simultaneously influence of the oxidative phosphorylation process to work simultaneously, and they are responsible for the death of that particular cell and in the influence of a specific inhibitor. So this is the experimental approach which is used for the titration, inhibitor titrations. Let me simplify this thing very well. So flux titration process is nothing actually. Gradually you increase the inhibitors and measure the flux. You did the titration, so you are adding something in some, and after some time you stop it because you get some color during the titration process. So same type of the approach is used here. Here you have to gradually increase the metabolites, the inhibitors, and finally you have to estimate the fluxes also. So this is the best thing, ATP synthesis and the mitochondrial oxygen consumption. So this is because flux you can only measure by either the product formation or by consumption of some components. So here we can measure this metabolic flux by the number of the ATP which are produced by this particular pathway. Simultaneously, how much CO2 is consumed here. That thing also. We have to generate the plot in between the flux versus the enzyme activity as we plotted previously. So here mathematical models are giving for the different type of the inhibitions. So if you are using the competitive, non-competitive, uncompetitive, so then you will find out the three different type of the metabolic control analysis we have to go ahead. So these are associated with them. Now the elastic coefficient is the third method which is used for the metabolic control analysis. Here we have to quantify the effect of the different metabolic concentration on the reaction rate. We discussed before that contribution of the different enzyme. Now we have to quantify the effect of the different metabolites, intermediate metabolites, final product, reactants, and what is their influence. So for that we divide all the metabolites of a biosynthetic pathway, degradation, whole biosynthetic pathway, targeted biosynthetic pathway, into the substrate specificity and the product specificity. So substrate, when we are considering the substrate for the as an elastic coefficient, so we have to use the E elasticity, and when you are using the product to understand the elasticity coefficient, then we are using the P elasticity. So they are the similar approach with respect to the product concentration, first one with respect to all the reactants. So this is the Michaelis-Menten equation which is the kinetics we are used, and approach is just at the low substrate. We starting from the low substrate concentration, and slowly then we increase and we decrease the substrate concentration, and we try to understand how this substrate concentration influence the different steps of that biosynthetic pathway. For cooperative enzymes, sometimes you identify an enzyme is working in many places. So for that, elasticity is very steeper, and a sharper flux you can find out as compared to the Michaelis-Menten. In the Michaelis-Menten equation, this is a hyperbolic, but here it is steeper because so many enzymes which are cooperative in manner, they are present here. So this is the same thing that I told you. Michaelis-Menten equation, that is the first one. Second is the P. First one is the, here there is a, how much they contribute in the enzyme reaction. As the substrate concentration is going to increase, so you identify that your reaction is also going to increase, and this is flowing the Michaelis-Menten equation. But if you increase the product, so you are going to understand that your product, when you are going to down, so your flux is also going to down actually. So these are the two equations, algorithms, which are basically used for this elasticity and the enzyme kinetics in the comprehensive way. The fourth part is the connectivity theorem. So this is mathematical algorithm. I am just explaining a few tips about this one. That is the local nature of the elasticity and system level relationship. So what is the local level actually? How are the different components are helping? What are the different parameters which are independent of the changes elsewhere in the pathway? So something which is independent of the pathway. So you have to consider those points, and those we can consider under the local nature of the elasticity. Second is about the system level elasticity. System level elasticity is about the flux control coefficient and concentration control coefficient. So these are the two different components you have to understand with respect at the level of the system. And whole is either the 0 and the 1 actually. So these are the two values you have to understand. It comes either the 0 and the 1. And the connectivity theorem describes this interconnected of a biochemical pathway basically. Connectivity theorem is helping to us now that how this biosynthetic pathway is connected with the another biosynthetic pathway, and what is the influence of the other biosynthetic pathway on this biosynthetic pathway. These type of the things can be go ahead by using the connectivity theorem. So this is the same thing you can identify. Elasticity coefficient was calculated from the Michelson models and for the Hill models also. The Km and the K0.5 value of the 1 millimolar in both the simulations, left coordinates and divided by 1/4 and right coordinate 1/4 position of the M actually. So that's why. So application of this relationship to calculate the unknown coefficient using known one. If you know a few relationship, you can calculate the unknown relationship also. Example is with the two enzyme actually. You have to start with the one enzyme system, then you have to start with the two enzyme system, then 3 and the 4. It's a laborious work. So you have to start the first one and you understand that it is something. Now question has come up that how you measure the FCC and the CC. So they are the direct and the indirect measurement. The first one we have to go ahead with the direct measurement. In the direct measurements we have to go ahead with the techniques. So first techniques is the inhibitors. You have to understand what is the influence of the inhibitor. What is the influence of the small RNA? What is the influence of the antisense approach? What is the influence of the oligonucleotides and the genetic modifications also? So you measure the knockdown via the qPCR. You can also make the knockdown by the CRISPR-Cas9 technology. You can understand this thing by the Western blotting and the enzyme activity ratio. Now the limitation of these things that sometimes if you are using the inhibitors, so you don't know, might be they have some unknown targets. So when they have the unknown targets, so it is difficult for you to go ahead with these limitations. Variability in the small RNA knockdown across the cell types is also measured. Indirect way, we rely on the elasticity coefficient only and the kinetic modeling. So challenges are many because at this the multiple allosteric modification and a factor in the dynamic intercellular environment is always a complicated situation and it is always a big problem. So sometimes we are making the link log models when we have some parameters are not available. So this is the way by which we can calculate the flux control analysis, and yes, for this we have used this reaction, and this is just like an illustration for all of you. I'm not going to detail, but yes, if you want to identify some inhibitor, some pathway, flux control coefficient, so we are using some inhibitor just like the rotinone, antimycin, carboxy, acetylosides and oligomycin, which act as a pseudo-reversible due to their high affinity for the target enzyme. So for the enzyme, for the irreversible inhibitor, some inhibitors are just like the common inhibitors, some are the irreversible inhibitors. So they can go ahead and finally you have to calculate this particular flux control analysis by doing this thing. And sometimes it is also our estimation. If you overestimate your flux control coefficient, so you lead to the inaccuracy in your biosynthetic pathway also. So now advanced development that come in this particular region is that development in method tailored for the non-competitive tightly bound inhibitors. So this is the improved equations it has so many different parameters which were not considered before, and it is used for the non-competitive tightly bound inhibitions. The benefit of this type of the work is that you tightly bound inhibitor with a great accuracy. You can go ahead with the great accuracy by using this tightly bound inhibitor. Secondly, others that you can utilize all the experimental data points for the very accurate flux control calculations. And application example are here, just like the mitochondrial respiration flux. If there is no inhibitor, inhibited the flux, inhibitor analyzed, just like these are the different inhibitors. Outcome is that better understanding of the distribution of the flux control on the mitochondrial pathway. Now the potential uses of this experimental approach. So we are using this potential uses. The first one is the oxidative phosphorylation flux analysis. We can go ahead with this one. So we focus on the analysis of the metabolic flux of the oxidative phosphorylation, this function in the various tissue. I mentioned this thing before a few slides also. So relevancy is help in the understanding mitochondrial pathology and their impact on the tissue functions. And key approach we can use only the MCA to assess the flux control distribution across the pathway and across the different tissues also. Now the mitochondrial pathology is another concept. Yes, characterize the some abnormality in the mitochondria, and that's why there is a oxidative, inaccurate oxidative phosphorylation. They disturb the oxidative phosphorylation. So it is linked to the specific mitochondrial DNA mutation affecting the whole oxidative phosphorylation, and finally they differentiate the different tissues. So tissue sensitivity factors which are mentioned here, that is the flux control of the coefficient of the mutated enzyme in the various tissues. And if you are working only on a specific tissue, so you have to use those specific tissue related enzymes which are only expressing their activity in those specific tissues. Then second, tissue ATP dependent and oxidative phosphorylation versus the glycolysis. So therapeutic uses of this oxidative phosphorylation is that can be used via the MCC. So tumor cell. Tumor flux control in the tumor cell is concentrated in the site 1 of the respiratory chain. So if you go and find out the respiratory chain, this oxidative phosphorylation formation of the ATP, it is very high, 0.3, and targeting site 1, this is the site 1. There are so many sites where this oxidative phosphorylation is carried out. So they tag the type 1. So site 1 is very selective, affect tumor cells without harming healthy cells with lower control in the site 1. So if you are working temporarily on some specific tissue, by using this method, so you can go ahead and you will target only those cells which are available there. So challenge in using the inhibitor in the intact cell, that is the hydrophilic inhibitor, poor cellular uptake, and hydrophobic inhibitors. And solution of that, we can go ahead with some long incubation with the careful maintenance of the cell viability that is required for this particular concept. So experimental determination of the flux control analysis, we can also go at this concept by using the objective, that is the quantifying control accepted by the pathways on the metabolic flux. This is the methodology, and this is the plot you have to plot, that is the generate the x versus flux plot, slope at the reference points, versus yield the elastic coefficients, and then producing the blocks upstream, downstream, elastic determined by the inhibiting the downstream enzyme, consuming blocks downstream, elastic defined by the directly from the system variation. So these are the really little bit elaborative, but yes, I'm just coming with the picture so that at least you know the basic of those things. Otherwise, we can come with the screenshot of the papers also. They will create the problem for you the better for the understanding. So understand here, this is the two block analysis. First is the producing block and second is the consuming block. So producing block means all those metabolites which are involved to produce that particular components. So whatever the factors which are used to synthesize that particular component, so we consider all of them into the producing block. Second is the consuming block in which whatever the metabolites you are looking, so where and how much concentration of that metabolite is consumed in the pathway. So elasticity is basically determined the inhibiting downstream enzymes, and this elasticity determined directly from the substrate variation. So this is the flux and this is the pathway you can show. It is going to up and it is going to down. So it is also considered with the connectivity forum, in which we also connect our biosynthetic pathway with the other biosynthetic pathway. So summation forum that come up in the final picture, that is the C1 plus C2 is equal to 1. But remember, it is never achieved the one target here. So this is the supply and the demand, just like the other components. So kinetics of the synthesis and the consuming blocks of the intermediary X. So one is the rate, second is the concentration. So it is going to, rate is going to increase and this concentration is going to decrease of the reactant. So this thing, rate plot of the supply and the demand block in a natural logarithmic scale. So this is the natural logarithmic scale in which we can plot it and we can understand very well. Now what is the application of this also for oxidative phosphorylation if you are using for this particular case that I told you, tumor cell, physiological insights, then case study ATP flux control. If you are using the ATP flux control now, not the oxidative phosphorylation. So ATP consuming blocks accepted 34% of the flux control in a particular cell. And if you think that if sometimes if you are blocking something by a single enzyme, even then those cells can survive. So it means there is some bypass pathway which is helping that cell to survive in those adverse situations with minimum activity at least. So similar control coefficient observed in the mitochondria with the ATP regenerating system. Implication is that you can validate the elasticity based FCC determination across the different experimental setups. So yes, this is the perturbation and the flux control. If you disturb the things, now depends on the enzyme activity. High control enzyme requires small perturbation for the accuracy. If you have the enzyme which is the highly in the controlled situation, so little bit small change is enough for the accurate estimation, accurate change in the metabolic control analysis. But if the low control enzyme you are targeting for your work, so they tolerate the larger perturbation with minimum errors. So practical observation that come up here, this control coefficient or the concentration depends and flux control shift with the pathway states. Example, in mitochondria, proton leak control decrease in the high flux states while ATP turnover increase. So whatever you know, this proton is moving from one membrane, inner, outer to inner, and that is the thing responsible for the ATP. But if you think the number of the proton which is responsible for the ATP synthesis, so might be the wrong because many protons are leaked by those membranes. So if you say that so much ATP is produced in the glycolysis pathway, so might be the wrong. It is less than 36, 34, 32. It is around 30 to 32 ATP which is produced in this particular case. So that is due to this thing. We have to consider those points also when we are considering this point. So these are the different kinetic models you have to use. I am skipping this point. So pathway modeling pe I will come here, how you model your pathway actually. So predict and explain system behavior under mathematical model of the metabolic pathway. So these are the different tools which you can use for the pathway modeling. The first one is the COPACI, metamodels, scan and the JSAT, CIS and SDML resources. So COPACI is very easy to use the tool for the metabolic modeling. So this is the link. Then metamodel is the advanced computational options. You must know some scripting knowledge, script knowledge to understand or to utilize this metamodel. Scan and the JSAT is very powerful tool for the synthetic biology. PISSEC is a Python-based flexible modeling. SMBL is a comprehensive link and the tool which you can use for the metabolic modeling. So if you are going with the SMBL resources, that's enough for you because all the resources are available here which you can use for pathway modeling. So now I am coming on the comparison of these different pathways actually. Structure modeling, kinetic modeling and the in vitro reconstructions. So pathway modeling are basically different from each other based upon these key features. And first one is the structure model, how you make the structure. Second is the kinetic modeling, how you understand the role and the phases of the different enzymes. And the third is the in vitro reconstitution. So for that, these are the key features, advantages, limitation and the application. If you go with the first one, if you have the structure modeling and pathway reaction structure based upon the psychometrics. So if you are trying to understand what is the structure of that pathway on the based of the psychometric ratio, so then you have to go ahead with the structure modeling. In structure modeling, what are the key features? That is the identified conjugate metabolites. You have to first of all identify the conjugate metabolites. You have to map the enzyme in the different groups also. And the third group, relationship. If you have the psychometric conjugate, if you know the conjugate metabolites and if you know the different groups of the enzyme which are used in that particular reaction, so then you can go ahead with the structure modeling. Advantage is that it reveals the experimental modules for the steady state relationship, provide a detailed pathway interaction maps also. Limitation of this structure modeling is that it does not allow the kinetic data actually. So that's why if kinetic data is not available, so detailed things you can't skip. But you can understand how many metabolites are involved in a particular biosynthetic pathway or degradation pathway. So application is that you can avoid the interaction and the relationship between the different metabolites in a particular biosynthetic pathway. Now I am coming on the second point, that is the kinetic modeling. So because the first structure modeling is not giving the enough information about the kinetics. So I am coming on the kinetics modeling. So kinetic modeling is the reaction kinetics. If you are focusing on the kinetics of the reactions, you are focusing on the enzyme activity and the steady state flux. So you have to go ahead with this particular concept. So you use the different rate reactions and equilibrium constants also, as we mentioned, include the enzyme elasticity and the product sensitivity also. And here, this simulates the flux variation. So there are so many simulation platforms available. You change the flux and try to understand what is happening here and facilitate in the in silico prediction of any pathway behavior. It requires the precise kinetic and the steady state data. It throws to the error from the incomplete parameter inclusion. Predictions, flux control and the system sensitivity, investigating therapeutic biotechnical targets. In vitro reconstitution is the experimental recreation of the metabolic pathway. If you recreate the whole metabolic pathway in some other system, then you allow the enzyme specific titration under the controlled conditions, ensure the reproducibility with the defined components, enable detailed analysis of the enzyme role, useful for the validation theoretical prediction also. And limitation is the limited by the enzyme availability and physiological relevance, difficult to maintain the substrate product balance basically by using the in vitro reconstitution. The application of this one is that it assessing the individual enzyme contribution to the flux, testing effects of the enzyme modulation. So we compared all the three different types of the modeling. You have first of all start with the structure, then come on the kinetics, and finally you focus on the in vitro reconstitution. So these are the modeling databases that I told you, SBML. It has all the resources. So you just click on this one and you can get much more information about all the resources. PISCES is a Python simulation for the cellular systems. So if you manipulate a few particular enzyme, so you can easily identify what will happen in the simulation situation. So this is the snapshot of the PISCES. Now, the in vitro reconstitution of the metabolic pathway. Now, there is a question, if you know a pathway, if you know the whole different, how you manipulate that pathway, how you control that pathway, so your another target is to in vitro reconstitution of the metabolic pathway. So objective of this in vitro reconstitution of the metabolic pathway is to identify the control exerted by the individual enzyme on the metabolic flux using the reconstituted systems. Methods are for the reconstitution of the pathway segments, assemble purified enzyme for a specific pathway of the segments. Titration, vary enzyme activity independently, measuring corresponding flux variation. And steady state flux monitoring is another concept you have to monitor, that is the quasi-steady state. These are the few kinetics related words actually. Maybe if you never heard, so it will be uncomfortable for you. But there are many books which can provide you enough information about synthetic biology and utilization of the LCA flux control analysis for to control any biosynthetic pathway. So this flux control thing is used to actually compute with the control coefficient for each enzyme. So application of the pathway modeling is that it can be used for the MCA application. Our target is to the MCA, and we know that how this pathway modeling is happening. What are the different structure modeling, then enzymatic kinetics modeling, and in silico modeling. So it simulates the single enzyme variation without disrupting the other components. Basically, you know what's happening when I know the case of some 2011. They told if you just manipulate a single biosynthetic pathway, so how could you believe that not other pathway are altered? How could you believe that this single enzyme is enough to change enough in a whole system so that it can give you the huge difference? So basically, we can take an example. Anything, whatever is coming from the outside, our cell is not easy to accommodate those things. If that component is increasing some metabolite, our cell does not allow them, because our cell has a very fixed behavior of the different metabolites and the different biochemical reactions. So how could you, if you are really achieved a high concentration of that metabolite, so definitely it gives some positive and the negative adverse or the positive effects on the different biosynthetic pathway. So that's why we have to do the simulates, different enzyme, different reaction. So we have to identify when we touch one part, change the one enzyme, so there is no change on the another pathway, and it is really going to increase the concentration of that final product. It also predicts the contribution of the different enzyme in the flux actually. If you know the contribution of the different enzyme in the flux, so definitely you can go ahead and you can control any metabolic pathway. So therapeutic insights, if you go, you can identify the control which are used for the drugs targeting. So for the drugs target, we know the different enzymes which are controlling the whole metabolic pathway. Then we can design some inhibitors, some activators which work on those particular targets, and they can inhibit and activate those biosynthetic or degradative pathway. So this way drugs can be designed. And identify critical control, then example is the same like the glycolysis pathway. If you identify a target which inhibits the glycolysis pathway only in the cancer cell or parasitic cell, so your whole job is done. So optimized pathway for increased production of the metabolite, that is the biophone and the pharmaceuticals. Now I am coming on the kinetic modeling applications. Kinetic modeling application is the second part. Here, after evaluation of the stability, robustness, and the dynamic properties, this model is really virtual. It itself is a virtual laboratory with parameters can be explored behavior under the diverse conditions. So these steps allow in-depth examination of the regulatory enzyme actually which control the whole metabolic pathway. So application in the pathway is the erythrocytes in the glycolysis. Here flux control mainly the PHK 71% and PFK 29%, and sucrose accumulation in the sugar cane. So we can control a few control points like invertase, glucose uptake, and the vacuolar sucrose transporter to control the sucrose accumulation in the sugar cane also. The another example is the glycerol synthesis in the cerevisiae. So predominantly it can be controlled by using the glutathione, this particular enzyme up to the 85%. And Calvin cycle we can go up to the 50% inhibitions of these two things metabolic. So this kinetic modeling is giving us the target to whom we have to manipulate actually. So this is the modeling glutathione and the PC biosynthetic for heavy metal resistance. We started our whole presentation with this illustration. So I am coming here. This flux control is the glutathione concentration. It is shared between the supply and the demand in both the stress and the copper stress condition, control as well as the stress condition, abatic stress condition in the presence of the copper. So traditional value is something different and views are different. Model inside are giving the different thing. Here, this is the rate limiting step based upon this location and the feedback behavior by the glutathione. But model inside emphasize us to go ahead about that. It is not only the sole rate limiting steps. It is also influenced by the supply and the demand. So glutathione demands plays a crucial role in the controlling the flux basically. So modeling glutathione and then here if we overexpress this particular enzyme, so it increase the glutathione, but we are not getting any increase in the flux. So cadmium exposure, when this cadmium comes into the picture, so this high glutathione demands basically high glutathione increase fluxes which is balanced between this and these two things. And our expression of the PCC without adequate glutathione transport can lead to the glutathione depletion and toxic effect also. So what is the purpose? We have to go simultaneously our expression of both the enzyme results in the significant increase in the glutathione and the PC control and the concentration. A twofold increase in both enzymes is enough to increase twofold increase in the flux basically. Previously it was the 30%, now it is the 60%. Try to understand. When a fivefold increase in the concentration of the both enzyme is enough to increase the up to the eightfold increase in the flux. So if you know the control points and if you are targeting all the control points simultaneously to improving the flux, so it can be easily achievable here. This is the things that I would like to explain here. This is the effect of the simultaneously our expression of both the gene encoding these two enzymes on the glutathione and the PC's concentration and the flux under the cadmium stress condition. So see here, that is the flux. It is going to increase here. It is going to increase this violet line and green line, while this red and the purple with very small amount of the concentration of these two enzymes are present. So that's why it can metabolites are present. So now I am coming on this model prediction for the inhibition pathway flux in the parasitic. So you can find out the different parasites, their K insights, their control points, targeting enzyme, flux control, inhibition for the 50%, flux reduction, and the drugs targeting the recommendation. So try to understand by if you know the metabolic control analysis very well, so you can manipulate those specific control points and you can target a few parasites also here. So this thing is possible. So this is the model prediction for the pathway in the parasites. So I will not go in the detail of this thing because it takes too much time, but yes, I would like to give a simplified version. If you are using the different parasites and if you know the control points, so you can stop their replication and stop their activity there. So these model flux behavior, when we inhibit the enzyme, so we can go ahead with this one. So genetic engineering is another concept to manipulate the different protein levels. So here we have to use the RNA antisense technology for the gene expression, attempt to modify the Rubesco, and these are the findings. So antisense technology is basically used for those genes which encoding those enzymes basically to overexpress the accumulation concentration of those enzymes so that you will get the better flux. Second is that Rubesco enzyme you can use to control the whole enzyme activity of that particular biosynthetic pathway. This modification to the Rubesco catalytic efficiency and our expression is basically unsuccessful to improving the photosynthesis process and some other related things. So DNA antisense technology was used to reduce the Rubesco activity in tobacco plants basically in 1988. It's an old story actually. So Rubesco is the limiting steps in the photosynthesis with control coefficient of 0.69 to 0.83 under high light actually, high humidity and low CO2, and it controls the decrease the significance under moderate illumination and the low CO2. So this is the limitation of the Rubesco modulation. You can understand this study did not measure control coefficient for the other enzyme in the Calvin cycle associated branches which may be significant. Conclusion is that Rubesco remains a K-limited steps under the specific conditions, but its manipulation for increased photosynthetic efficiency has proven till today challenging. So that's why Rubesco, how much you manipulate, because it is not only the one enzyme which is playing the critical role, some other factors, control coefficients are also involved. This palliatopic effect and the model validation, palliatopic effect is the PFK1 downregulation leads to decrease the activity in other enzyme affecting the flux control. complicatory approach in silico modeling. We have to predict the flux control steps while in vivo experiment validate these predictions, providing a more accurate understanding of the pathway behavior. And comprehensive approach that we have to use combining in silico modeling with in vivo experimental, they can improve the metabolic control analysis very well. Accurate flux control coefficients can be determined and unexpected effects can be identified for the better pathway optimization. So this is the fine-tuning cellular protein expression. So fine-tuning, as I told you, we are trying to express the protein in another organism. So limitation is that knockdown experiment often focus on the high inhibition up to the 80% or wild type layer, overlooking the intermediate layer. So sometimes we don't want to knock down a particular enzyme up to the 100% or 80%. We want to knock down into the 20%, 40%, 60%. If you know, so gradually you can easily understand the whole metabolic control analysis. This limits the ability of the obtain the reliable control coefficient basically over a wide range of the enzyme activity and the fluxes. And second is the overcoming limitation with the inducible gene expression. Inducible system is systematic system like the lag, lambda, tetracycline promoters in bacteria and the yeast allow control of the gene expression. Problem is that difficult achieving a steady state of the protein expression. So now synthetic promoter library are synthesized that can give the different transcription strength for the synthesis of the different number of the mRNA which is associated with a particular enzyme. Promoter enable precise control over the protein expression both above and below the wide type level enhancing MCA investigation. So application of this synthetic promoter library is very well. It can be easily used for the metabolic control in the E. coli and this lactase. This is also used to determine the glycolysis control in the E. coli and lactase. Developments in the application of this SPL, it is being extracted to the yeast, mammalian and the plant cell. And advantage of the genetic engineering with the MCA provide the powerful tools for optimizing the organism of the biotechnical interest. Advantage is we know the pathway structure very well, independent the manipulation of the enzyme concentration and the effectors, and third one is the validation of the theoretical model. So limitation of this particular investigation is that dilution reduce the enzyme-enzyme interaction many times, and so that whatever the enzyme activity you are monitoring, it will also reduce down. Quasi-standard state condition may not fully mimic in vivo dynamics. In vitro it is possible, but in vivo dynamics, this quasi-state condition is not possible. Limited commercially available of the purified enzyme is also a major challenge for this SPL technology. Now the case study and the finding, Krebs cycle, we work, many persons work by using these upper glycolytic pathway, lower glycolytic pathway, in which they use the different promoters, and finally they identify the different control coefficients. So applications of the therapeutic, that is here, complementary analysis is also mentioned here, in which elastic analysis and the in vivo validation ensure the comprehensive understanding. Incorporate advanced techniques like real-time metabolomics. We have to ensure to get the refined data actually. In vitro reconstitution, that is a very powerful tool basically for dissecting the pathway control, and it provides the detailed insight into the enzyme level regulations. And the limitation addressed by the advances is that recombinant DNA technology enable the broader applications, integration with other experiments and modeling approaches ensure the robust conclusion also. This is the conclusion of this metabolic control analysis. Basically, it provides a simple framework for analyzing the control coefficient, means control distribution in any metabolic pathway. It is useful to design the experiment to investigate the pathway dynamics. The key insight from the NCA is that this flux control is typically distributed across the pathway, not concentrated in a single step. Please take care of this statement always. Distinction between the control and regulation is crucial. So one is the control, second is the regulate. So these 2 points are totally different from each other. So you have to differentiate which steps is regulate, which step is the control, and how these are the control and how these are the regulate. Feedback regulation is often decrease the flux control coefficient of the regulated enzyme, contrary to the expectation of the increasing control. Feedback stabilize metabolic concentration also. So this is all about this metabolic control analysis. Yes, many thanks to all of you. And yes, I would like to acknowledge again one of my students, Sachit, for helping me to make these PowerPoints. And I would like to acknowledge the GYAM and the Ministry of Education to fund this GYAM course along with Professor Jain Jo. So if whatever the question you have, you can ask. Yeah, it's only 4 minutes, 3 minutes left before 5 p.m. So we would like to conclude this session at the 5:10. After 5:10, you will go in the CIL and you check your NMR and you get a chance, so you can request them also for the HPTLC. So yes, thank you all of you. Whatever the question in your minds you can ask, because this is a tutorial, and whatever the question related to your work you can ask, and with respect to this course also you can ask, because tomorrow we don't have enough time. Our schedule is very tight. We start early morning and we have to free all of you just during the lunch time. So if you have any doubts regarding your credits and any other things, so we are available. So we try to use your accounts, ABC accounts, okay, and we shift those ABC accounts, your Aadhaar number, ID we also require. So we would like to request each and everyone, please collect one person, I will depute one person, so he will write down your Aadhaar number and your name, correct name, as well as your ID, so that we can transfer this one credit to your ID. This is the first thing. And second thing is that these PowerPoints you will find out very soon, means only after tomorrow. These PowerPoints Ritu will give you in the morning to all of you, but we have to arrange them systematically so that you can arrange. Otherwise, randomly we can give it in the 2 minutes. The third point is that this is a course that is present in the SWAM also, and it will be forever with the ministry. So we have to try to maintain all the records up to 3 years of this thing with us. And it is a very fixed schedule. Yes, many things we left because it's very difficult to cover the whole things in the 12 lectures of the whole course. So we can give you the overview of the metabolomics and try our with best split, so that you know a little bit about the metabolomics and how it can be useful by using the different techniques, how you collect the data, how you generate the data. And tomorrow I have a class in which I request you to share the dais with this platform with me, and she will explain the data. What type of the data she generated, first thing. Second, what are the raw data of those data she compiled in a paper, and what are the pipeline between the raw data and the final data she published in a paper. So these three things of the four different technology. First is the GCMS, second is the LCMS, third is the NMR, fourth is the FTIR. So she explained these things within the 30 minutes. If she is not able to explain, so I will be with her here, so I will explain those things as well. And we try to both, but the 10 to 15 and the 20 and the 30 minutes, I will take the same topics, that is the plant disease and the metabolomics. So the first part is the plant disease and the metabolomics, and the second part is the data interpretation of the plant disease and the metabolomics. So this way our whole 1 hour is complete in the morning. So in the morning it will be my class, and I think schedule is at the 9 a.m. right now, because we have to cover the 4 hours before the 1 p.m. actually. Every day in the GYAM, it is mandatory to complete the 4 hours at any cost. So today we started a little bit late in the afternoon. It was the 1:20. So we try to complete our 2 hours at any cost, because everything is the recording actually. So yes, whatever the question in your minds you can ask, departure schedule you can ask, so that we can help you in those matters. And for this ABC accounts, I will make a paper, I will give you the column. You have to write down your column very well, or we will share a Excel sheet. You write down your ABC account, your name, your email ID, again, as well as your other things, information. so that we will transfer to the examination branch. And yeah, बोलिए या क्या करें बोलिए.
सर एबीसी अकाउंट क्रिएट हमें खुद करना पड़ेगा.
हमारे पीछे बच्चे बैठे हैं, इन्होंने अकाउंट क्रिएट किया नहीं किया हुआ. इन लोगों के पास में एबीसी अकाउंट है. तो जिन लोगों के पास में एबीसी अकाउंट नहीं है, तो आप पीएचडी स्टूडेंट हैं, आपको भी कोर्स वर्क करना पड़ेगा. तो अभी एबीसी अकाउंट में यदि आपका क्रिएट होगा तो उसमें ये ऑटोमेटिकली चला जाएगा. तो आपकी जो फाइनल वहाँ से सर्टिफिकेट रिलीज होगी उसमें ये आपको मिलेगा एक क्रेडिट. तो ये जो हमारे जूनियर बच्चे थे हमने उनको लिया नहीं, सिर्फ 2 बच्चे सिलेक्ट किए हैं. इनका 2 क्रेडिट का मोक कोर्स होता है. 1 क्रेडिट तो उनका यहीं से आ जाता है. If you do the other book, so your 2 things are completed. So this thing can be done in this way also. So this is just part. This 1 credit is a part of your curriculum, and it will become automatically. It will come in the जो आपकी वहाँ पे database है, नेशनल लाइब्रेरी database में भी हमारे सारे lectures अवेलेबल होंगे, and you can 1 कॉपी की सारी वहाँ पे डिपॉजिट होगी. तो वहाँ से भी आप ले सकते हो. उसमें ये भी डिटेल होगी कि किस किस ने इसको अटेंड किया है. हम आपकी सारी डिटेल्स वहाँ पर शेयर करेंगे, सो दैट दे कैन ट्रांसफर इन द फ्यूचर टू यू डन दिस पर्टिकुलर कोर्स हियर. सो एनी इंफॉर्मेशन यू वांट टू टेक केयर. या मोनिका, could you help them how ABC account is created? They come here and speak. Yeah, now.
Just need to search ABC ID login on Google. Sir, it's okay. My voice is already too.
Yeah. So now you can open it. See helping how to make your ABC accounts. If you don't have, please make your ABC account because it is necessary for us. Yeah.
Search ABC ID login on Google. And then you will get first bar like ABC Academic Bank of Credit Test. Then tap on the link.
सर्च करते हैं ABC ID लॉगिन, तो ये पहली लिंक आती है जो abc.gov.in की. तो उसमें अगर हम क्लिक करते हैं तो ओपन हो जाएगी वो. तो आपको ऐसे कुछ इंटरफेस दिख जाएगा. तो इसमें आइदर आप डिजीलॉकर से लॉगिन कर सकते हैं, और अगर आपके पास अकाउंट नहीं है तो आप यहाँ से साइन अप भी कर सकते हैं. लॉगिन पे जाएंगे तो स्टूडेंट पे क्लिक करने के बाद वो आ जाएगी आपको साइन अप की ऑप्शन. जैसे कि मेरा ऑलरेडी बना हुआ है तो इसलिए मेरी डायरेक्ट आ जाती है. तो मैं इसको लॉग आउट.
डॉक्टर साहब ये आप लोगों का होगा ना, तो इसमें कोर्स अपग्रेडेशन में आएगा आपको.
हाँ, तो मेरे लिए हमारे लिए आवर्स कैलकुलेट होते हैं. जितना आवर्स मैंने यहाँ पे लिया है वो बाई 10 हमारा वो आ जाएगा, स्कोर आएगा. एफडीआई में हमारा स्कोर वैसे आ जाएगा.
एटीआई स्कोर है सर.
उसमें आ जाएगा.
एटीआई स्कोर में
वो प्लीज सीआईएल में कौन लेके जाएगा इनको. सीआईएल में नीचे रिक्वेस्ट आप लीड कीजिए, और आपको कुछ फोटो सीआईएल की लेनी पड़ेगी, है ना. आपको इनके साथ जाके वहाँ कुछ फोटो क्लिक करनी पड़ेगी. वहाँ पे इंस्ट्रूमेंटेशन देख पाए तो 10-15 मिनट लगेंगे. वहाँ फोटो क्लिक करके फिर आप लोग कर सकते हैं. तो प्लीज एवरीवन यू हैव टू गो एंड अंडरस्टैंड द एनएमआर होता कैसे है और वो कैसे किस तरह से डाटा को रिजल्ट करता है. सो दिस इज माय हम्बल रिक्वेस्ट. ले जाओ.